Thank you. Uh, Hi, everyone. Welcome to this month's MCM. Today, I have the pleasure of introducing Dr. Michael Treadway. Dr. the Winship Distinguished Professor in Psychology. He's also the clinical director or the director of clinical training at Emory University. He's a clinical psychologist and neuroscientist. He got his PhD from Vanderbilt, followed by his internship and postdoc at Harvard Medical School and McLean Hospital. He's now the director of the Translational Research in Affective Disorders Lab, the TRED Lab, which studies the behavioral and neurobiological determinants of cost-benefit analysis, decision-making in healthy individuals, as well as in patients with mental illness. Dr. Treadway's lab uses a plethora of techniques to better understand the molecular and circuit level mechanisms of psychiatric symptoms related to mood, anxiety, and decision making. He's received numerous awards, such as those from the APA and APS for early career contributions. And today we are really fortunate to have him here about effort-based decision making and its discontents. Please join me in welcoming Dr. Treadway.
Great. Thank you so much. Thank you, Colleen, and all the organizers so much for the invitation. I think this is a great speaker series that I've really enjoyed. It's a pleasure to get a chance to present some of our work. Um, when I received the and I asked the organizers which of some various ongoing activities in the lab might be of most interest, and they encouraged me to present on some work focused on trying to understand different subcomponents or subtypes of anhedonia and then the potential relationships to inflammation. So that's really going to be the focus of today's talk. I'll begin just by acknowledging some industry relationships. None of these had any role in funding or really at all involved in any of the work I'm going to present today. So, as Colleen mentioned in her introduction, we've been very interested in understanding sort of cost-benefit decision making, and particularly how it is affected in a symptom common to depression, but by no means unique to depression, referred to as anhedonia. So, in the context of depression, anhedonia is defined by the DSM a lack of interest or pleasure in previously enjoyed activities. And it's highly prevalent. It can also occur in the context of schizophrenia disorders, anxiety, stress, and trauma-related disorders. And one of the things that our group and a number of other groups have highlighted over the last really 15 years now is that this definition seems to draw a false equivalency between losing interest and not experiencing pleasure in response to activities. And that those are from a nosology perspective, as things currently stand, those are basically treated as the same problem. And there's now been really 2 decades worth of behavioral and human cognitive neuroscience arguing that, in fact, those describe changes in systems related to, colloquially either wanting or motivation or craving or willingness to expend effort to pursue something versus the capacity to like or enjoy or experience pleasure when something positive happens. And there's been a lot of work to suggest that these are neurobiologically dissociable systems, and therefore it seems reasonable to hypothesize that at least for some individuals, they might have a primary problem in one system, but not the other. That's been a longstanding hypothesis. There has been, however, challenges in finding, I'd say, robust evidence that this difference is really clinically meaningful at the level of the day-to-day lives of patients. So, sometimes when I've presented my work over the years at more clinically oriented conferences, I get pushback from psychiatrists and clinical psychologists sort of practicing in the field, basically arguing, you know, maybe this is true at the level of fancy neuroscience, but when push comes to shove, people like what they want what they like, and those co-occur so often that I'm not sure this is really a meaningful distinction. And so one of the things we wanted to do in the first set of studies I'm going to present is to see if we couldn't find evidence for really different dynamics between sort of a primarily what we refer to as motivational anhedonia or a deficit in wanting versus considonia or a deficit in the ability to enjoy things. And so this first set of data I'm going to present on, this has not been published yet. This is all relatively new from our lab. It's really led by the lab's assistant director, Jess Cooper, as well as a research assistant in the lab, Tatiana Pillsbury. Um, so, as I said, this has kind of been a long-standing idea for at least a couple of decades. And during those decades, there have been a number of different sort of clinician report assessment instruments developed to try and tease apart motivational and consummatory anhedonia. One that we study is the motivation and scale. It's a self-report version adapted from a clinical interview called the Canes that was initially designed mostly in the context of negative symptoms to these sort of motivational deficits versus consummatory deficits. And there are two subscales of this instrument that focus on these two different domains that I'll just refer to as sort of MA and CA throughout the talk. Other instruments have been developed as well. There's the original TEPS from David Gard. There's the Dimensional Anhedonia Rating Scale, the DARS. And all of these have been to tease these apart. They all have this sort of core problem uh, of a cross-sectional assessment at a single time point, which is the two subscales tend to be extremely highly correlated. All right, so this is data. This is the correlation from the data set I'm going to present on. We have about 166 participants. About half have clinically significant anhedonia determined by SCID, and as you can see, the correlation is I think about 0.78. So they're not isometric constructs, but there's a lot of overlap. And what we wanted to do in this study is see if by using sort of daily EMA surveys, we could determine whether the trajectories of within subject responses to effort and reward led to suggested that there was this sort of variation here, high motivational anhedonia, but quite a bit of variance in your score in consummatory anhedonia, for example. Is that actually meaningful? So our protocol for this study is we recruited people to the lab. We brought them in for clinical interviews. So everyone was assessed by SCID and completed some self-report questionnaires. This data set was aggregated over by combining several ongoing trials in the lab. So the amount of EMA surveys, the number of weeks they did vary a bit based on what study we were pulling folks in from. That was always included as a covariate. But after their clinical interviews, they then did EMA surveys for a number of weeks. And our EMA protocol began, it was 6 surveys a day. The first survey was always at 10 a.m. And we always begin by kind of asking people to rate their momentary mood using items drawn from the PANA scale. And then we asked them to tell us, so what do you think you're going to do in the next couple of hours? They have a drop down of about 18 activities they can choose from, or they can work if they're in. Um, and then how effortful and rewarding they think that activity is going to be. Then after time passes, 2 hours later, time for the next survey, they tell us, did you in the prior survey, you said you were going to, for example, watch TV. Did you do that? If so, tell us how effortful or rewarding it was. So we can then kind of compare what they expected versus what happened. And then, and tell us your affect and tell us what do you think you're doing in the next couple hours, and this so on and so forth repeats throughout the day. So again, 6 surveys per day. So in the data I'm going to present, we had 163 participants, about half, 84, a little more than half were healthy controls. 57 were anhedonic and met full criteria for a current depressive episode. 22 were anhedonic but sub for MDD. Um, and they did EMA surveys, like I said, between 1 to 8 weeks, 6 times a day, every other day. Um, we also, for all these activities, we wanted to get a sense as to how these activities were viewed generally. So we recruited an independent sample who just rated these activities in terms of their average effort and reward. 102 online participants. And this was done to have a way of differentiating between someone who's maybe doing a normally enjoyable or relaxing act, but because of an anhedonia process that they themselves are not, versus someone who's reporting not enjoying something because they're doing it really enjoyable. So these ratings provided a way to look at difference the types of activities that people were engaged in terms of their effort and reward levels. And so I'll begin presenting on relationship to these sort of more objective or independent ratings, and then we'll get into the subjective assessments. So the first kind of question we had is just in terms of the activities that people with relatively higher motivational anhedonia scores, as assessed by the MAPSR versus anhedonia scores, do they show sort of different patterns in terms of the mean effort rating or reward rating for the activities they spent their time doing over the EMA period? So these are just simple linear regressions. I'm sort of showing you the model up at the top. And looking at motivational anhedonia in terms of, again, these independent rating, effort level ratings of activities, we see, as you might expect, sort of almost like a sanity check, that individuals were, with higher motivational anhedonia are engaging in activities that are rated as generally less effortful. So there's sort of avoiding effortful activities, one might say here. Now, in contrast, individuals with high consumatory anhedonia really are not showing this pattern. And if anything, they are engaged in more effortful activities overall, which for us was, a striking initial example of these two sort of subtypes of anhedonia not necessarily behaving the same when you're their activities unfolding in daily life. And this, I should note, the scatter plots here are being shown for the planned activities. So on every survey, when what are they doing next? Pattern looks very similar if I plot it out as a function of the completed activities. And I'll also note for every activity, we asked participants to indicate if they felt they had control or not over what they were doing, right? If, could be that maybe you're engaging in really unrewarding activities because you don't feel you have any choice. And there were no associations with MA and CA and mean level of control, and including control as a covariate doesn't change this relationship. As an aside, I've also been, long interested in, distinctions between the sort of anhedonia domain and more general measures of internalizing psychopathology, symptom severity. So just staying with the same mean effort ratings, if you look at other standard measures like the BDI, the GAD-7, or the SHAPS, you see that this is not just tracking with all, broad internalizing psychopathology severity. The SHAP starts to get a little close, which makes sense given that it's more focused on the anhedonia domain, but clearly not as strong a relationship, a subscale focus specifically on motivational anhedonia. Turning to reward, same question. Here, we see actually individuals with higher motivational anhedonia are choosing more objectively rated rewarding activities. This was a bit of a surprise. And when we dug into the activities that were sort of driving this, they tended to be sort of low effort, potentially high reward activities like social media, watching TV, playing video games, and uh, they so it's sort of it's almost like a kind of still an effort of aversion and gravitating more towards almost like comfort food type of activities. For individuals with consummatory anhedonia, we definitely don't see that. So here again, when looking at what people are doing, we're seeing these subscales kind of map on in the directions we'd expect. And again, very similar if looking at completed versus planned activities. Um, Now, moving from these sort of objective ratings to sort of people's more subjective experience, we want to see if we could understand how motivational and consummatory are related, in which the effort and reward of what people are doing influences their affect. So again, at every survey, we would ask people to rate their affect. And when they're making that rating, the things that might be influencing their affect that we could measure could be the influence of the activities they had just completed. Maybe they just had an exam, for example, and that was really effortful, and that's influencing their affect. It could be related to a sort of prediction error related to what they just completed. So maybe the exam to be hard, but not too hard. And it turns out it was super hard. And so it's sort of big negative prediction error there in terms of the effort and mapping their affect. Or it could be the what they are looking towards next, that, the exam's over, I'm done with it. I'm really looking forward to hanging out with my friends next. And that's actually what's driving my affect. So we a series of models with all these components combined using linear mixed effects models. Um, and so we have about a total around 6,500 surveys for these models. And what we're looking for is for predicting positive and negative affects separately. Did the levels of or rated, subjectively rated reward or effort or prediction error of reward or effort of the prior activities or the reward or effort of the upcoming planned activities, how did those predict positive affect over participants, and what were the interactions of those terms with either motivational or consummatory anhedonia? And I'm just going to, a few results we have so far. This is still an ongoing project. Really, there was a significant interaction between motivational anhedonia and the subjectively rated prior effort of a prior activity on positive affect, such that actually individuals who were at the highest level of motivational anhedonia had a slight increase in mood when they fall, which was a distinct pattern for people with sort of lower levels of anhedonia. And I think this maps on really quite nicely to what we might expect to see from behavioral activation therapy, which is often trying to help people improve their mood by getting them to engage in activities that they've been avoiding in part because they feel too effortful. This is sort of illustrating that, yeah, individuals with motivational anhedonia, they do tend to respond to and feel a bit better when they exert effort. Now, interestingly, this pattern was really not the case for individuals with consummatory anhedonia. And in for those individuals or with higher consummatory anhedonia, doing something that was really effortful was really associated with a significantly worse level of positive affect. When we looked at the influence of prior on positive affect, we saw, as expected, it had a mood elevating effect, but there was really no interaction for either anhedonia subtype there. And a few additional results I'm just going to walk through very quickly in the interest of time. Um, we did see an overall positive effect for reward prediction error, but not effort prediction error on positive affect, but no interaction with MA or CA. And we didn't see any interactions with MA or CA for reward or effort predicting negative affect. Um, and we didn't see interaction between MA or CA for planned activities, only, as I presented earlier, prior activities. So just to summarize this first part, I do think this is some compelling evidence that motivational and consummatory anhedonia do really manifest differently in daily life. Particularly individuals with high MA show a kind of behavioral activation response pattern of responding with higher positive affect when they actually do something effortful, while those with consummatory anhedonia do not. And this may have relevance for thinking about who may be a best responder for that type of behavioral intervention. So, having spoken a little bit about the difference between MA and CA in terms of sort of symptom experience, I'm now going to kind of to the level of neurobiology and talk about some of our work suggesting that one specific pathophysiology for motivational anhedonia may be chronic inflammation. So inflammation has become, I think, kind of a hot topic. When we're talking about inflammation in the literature, usually what people are referring to is evidence for increased inflammatory markers, suggesting that the innate immune system is engaged or more active than might be expected, given the absence of an acute infection or other disease process. The observation that patients with depression, although not uniquely depression, but certainly it's sort of this started in depression, um, that patients with depression have elevations in inflammatory markers goes back to the early 1980s and has just been repeated over and over again in multiple studies and meta-analyses. Here's an example from a recent meta-analysis highlighting the increase in different markers. The green arrows indicate some of the most commonly and reproducibly observed increased markers, including IL-1 beta, IL-6, TNF alpha, and CRP, C-reactive protein. Another thing that this particular meta-analysis did that I really like is they looked in all their studies that were included in the meta-analysis and asked the question for all the patient samples, what percentage of the depressed patients met CDC guidelines for having high inflammation, right? Do you get these meta-analytic results because all depressed patients are a little inflamed, or is it really driven by a subgroup? And this is the funnel plot for that result. And what you can see is it's really about between 25 and 30% of depressed patients across all these samples who are showing this elevated inflammation per CDC guidelines of a CRP greater than 3. So that sort of suggests potentially a subtype, and that not all patients meeting criteria, in fact, not even most meeting criteria for depression, are going to have high inflammation. Another strong set of cross-sectional evidence came from the UK Biobank. It's a study of around 30,000 individuals, and they were looking to see, okay, in depressed patients, they were looking across a range of conditions, but in depressed individuals, what does elevated CRP correlate with? And the number one hit was feeling everything was an effort. There was also some disruption to appetite, and then also could not get going, loss of energy. So these are, sort of key descriptors of what we might consider motivational anhedonia. And that's sort of cross-sectional evidence. It's as far as it goes. Where I think the case for inflammation started to really get interesting and compelling was from the original interferon alpha studies. Interferon alpha is a synthetic cytokine that was used to treat cancer and hepatitis and causes a massive engagement of the innate immune system. And what people began to notice in the 80s and 90s is a subset of patients receiving this drug would start to get really depressed. And my collaborator here at Emory, Andy Miller, ran a study, this is about 25 years ago now, where they recruited a sample of individuals who were about to begin treatment. They screened them. They only included individuals who had no history of psychiatric disorders at the beginning of their interferon alpha treatment. And remember, this is a life-saving treatment, basically, that they're about to, and despite that, and despite the fact the treatment was succeeding in addressing its target, about half of this sample became depressed after 12 weeks. And I think it's extremely interesting that you can take individuals who've never been depressed before and you can induce a major depressive episode simply by manipulating inflammation. And it's also interesting that it only happens to some people. So I think this our core idea of what the inflammatory subtype is it's not the mere co-occurrence of depression and inflammation. There's a subset of people who, when experience a lot of inflammation, have a sort of vulnerability with regard to developing motivational deficits. That, as you can see, some people in this study they had real high inflammation from the drug, but they weren't necessarily getting depressed. So what inflammation is doing, why might it be leading to sort of inflammatory, to motivational deficits? Another study by Andy and his colleagues sort of tracking interferon alpha patients used dopa PET imaging before and after interferon alpha and found that interferon led to a acute reduction in dopamine availability and synthesis capacity as by um, FDG, F-dopa PET, excuse me. More recently, our group working with a molecular biologist named Zhixing Wen here at Emory has used an iPSC model where we cultivate human dopamine neurons in 2D culture and have dosed them with IL-6. And here you can see vehicle, this is electron little vesicles that contain dopamine in dopamine neurons. This is what will go on to release dopamine when the neuron fires. And under IL-6, the number goes way down, and the dopamine concentration within each vesicle goes down. And then you can reverse that effect if you co-administer IL-6 and an IL-6 JAK-STAT antagonist called baricitinib, which blocks intracellular signaling along the IL-6 pathway. So these dopamine neurons are responding directly to cytokines, reducing their level of dopamine. And you can also down here see you get real IL-6 induced changes in the level of firing rate. So there is, I think, good evidence. It's certainly not the only thing that inflammatory cytokines do to the brain, but there is good evidence that they are really changing how the dopamine system functions. And what we know, and I'm sure this group is very familiar with, is one of the things that will change if you really reduce dopamine availability is you'll change motivated behavior. This has been demonstrated. There's a set of studies from John Salamone's lab showing with microdialysis in rodents that response to IL-6, you get this real reduction in extracellular dopamine levels. And in these same animals, IL-6 leads to a real dose-dependent decrease in lever pressing for reward. Looks very similar to what happens if you administer a dopaminergic antagonist. A few years ago, we did a review sort of demonstrating responses to acute inflammation manipulations across different species. And you tend to see this same phenomenon across model organisms of, measures of dopamine function drop and measures of effortful behavior, willingness to work for rewards tends to drop as well. So, given this, can anti-inflammatory medications be antidepressants? And this is where I think this line of work really ties back to the first half of the talk. A trial was run here at Emory in 2013, trying to see could we use a powerful anti-inflammatory, a TNF-alpha antagonist called infliximab, to treat depression. And looking at overdepression severity in a 12-week trial, you got absolutely no separation from placebo. I mean, completely dead. So then what they did as a post hoc analysis is they went back and they identified the subset of patients who had high inflammation at baseline. And there you did see in red versus blue significant reduction. And importantly, the symptom with the highest reduction was the work and activity symptom from the Hamilton rating scale for depression. They unfortunately in this early trial didn't have measures that were more focused on motivational anhedonia. A second large study, this one run by Johnson and Johnson, they tried sirukumab and an IL-6 antagonist. Now they did in this study only include as an inclusion criteria patients with evidence of high inflammation at baseline. So they were trying to learn the lesson of the prior study. Let's use our drug just focused on those patients. But again, you see overall depression symptom severity. You did not get any separation. However, when they looked at a more anhedonia specific measure, the SHAPS, they did start to see a signal. So the goals of the study I'm going to present on today is to sort of building off X. We wanted to see if you saw clear evidence for an effect of an anti-inflammatory drug. If you looked in patients with depression and high CRP, and you used a sort of rich characterization of motivational deficits with a variety of both task and self-report measures. And importantly, we also excluded extraneous sources of inflammation from the samples. So, you know, again, just because someone is depressed and happen to have high inflammation, that doesn't mean that inflammation is playing any kind of causal role whatsoever in their depression. There's a vulnerability we believe some people have to inflammation. We don't, as of yet, as a field, know what that is or how to measure it. So we will recruit patients with depression and inflammation and no clear, obvious cause, like an autoimmune condition or an active infection or an uncontrolled medical condition for that inflammation, with the idea that that is enriching our sample to include people who have this vulnerability. But even there, not everyone in our sample will have the vulnerability. This was our study design. So we went through screening measures, and then they did a baseline session with self-report behavioral tasks, imaging. They then got randomized to a single infusion of either infliximab or saline, and then came back 3 days to do some measures, and at 2 weeks to do sort of repeat all the tasks and imaging they'd done at baseline. And we did it at 2 weeks because we felt that was the longest we could wait to allow the drug to have an antidepressant effect while still being confident that the drug would be actively inflammation. After about 2 weeks on a single dose, inflammation can start to creep back up. Our primary behavioral measure here is an effort-based decision-making task, very similar to many others that have been used in the field. This one's adapted for the imaging environment, was done in the scanner. Basically, it's a series of accept or reject choices that people make in response to varying levels of effort. Effort here is physical effort in the form of button pressing, set to either 20, 50, 80, or 100% of someone's max press rate for 20 seconds. And so in each trial, they're choosing, do you want to do this much effort in exchange for, say, in this trial, $3 reward, accept or reject to the next trial? One that I think is different about this task as compared to other tasks is they are not doing the effort as they go. They make a bunch of choices outside the scanner and they complete the effort that they chose. And I think that matters because it means that sort of as people are going through the task, they're kind of accumulating a bit of an effort debt, which if you think that there's an aversion to effort, that might strain more heavily on people who are really sensitive to effort. And we people's choices using a sort of ending model. This is data from a set of healthy controls, just sort of showing that kind of effort discounting. Before we broke blind on the study, we pre-registered our primary outcomes. So very quickly, we expected inflammatory would go down with infliximab relative to placebo. We had several self-report questionnaires that we had pre-registered as sort of measures of motivational anhedonia, where we expected to see a group by time interaction there. We expected infliximab would lead to an increased willingness to work for rewards on the EBDM task. And then we expected it would change subjective evalu encoding in ventral striatum and DMPFC with our imaging data. So what we found in this is first, as expected, infliximab did a nice job suppressing CRP. We looked across inflammatory markers and we saw that you got a fairly decent size group by time interaction effect. That's the partial ETA squared interaction effect on the y-axis here. The strongest effect was for the soluble receptor TNFR2, which makes sense given that it's the soluble receptor that really lies directly on the pathway that the drug targets directly. So we used this as our metric for sort of target engagement or effectiveness of the infusion. Looking at sort of our self-report measures for motivational anhedonia, these results were a little mixed. We did get a strong signal in the infliximab group for two of the four in terms of improvement. We also did get a somewhat of a placebo effect. And so we didn't, even though we got nominally stronger effects for the infliximab group, we did not get a full group by time interaction. I think one of the challenges here is anytime you do a more invasive intervention study, like there's brain stimulation or there's an infusion or a surgery, that tends to lead to quite significant placebo effects. And I suspect 14 days was not enough time that really wear off. And so we didn't get that separation. Looking at our behavioral task, which is arguably a little more state dependent, so more just a reflection of where people were at 14 days. Here you can see at baseline, there were no group differences. You see sort of a main effect. People choose fewer effortful options as the amount of effort increases, as you'd expect. After 14 days, however, we did get a significant group by time as well as a group by time by effort level injection, such that individuals receiving infliximab chose more effortful options, and the differences were most pronounced at the highest levels of effort. So they were really more willing to work, particularly at those upper levels of effort, which is what we'd expect to see if we're improving motivational anhedonia. Looking at effort discounting curves, you sort of see a very similar pattern here. So at baseline, no difference. And at 14 days, sort of clear separation between their discounting curves. Moreover, both of these metrics, either choices or the model-derived discounting parameter, sorry, it keeps being a delay in there. They both tracked with the level of change in TNFR2. So, what you're expecting to see, particularly for those who received infliximab, is TNFR2 goes down. And you can see for the people for whom it went down the most, they also increased more in their expend effort or or decreased in their effort discounting. In the interest of time, I'll highlight sort of one imaging finding. So, our idea here is that one of the things that happens when you reduce inflammation is you should sort of restore some dopaminergic tone. So we thought that even though fMRI is not a direct measure of dopamine by any means, that shift might be reflected in more stronger subjective value encoding in the ventral striatum. We did not, however, see any univariate amplitude change in striatal activity in response to a sort of subjective value parametric modulator. We did, however, also try a multivariate analysis, which may be a little more sensitive, particularly for sort of longitudinal analysis. And they're using a k-fold cross-validation with a PLS classifier. We did see that the pattern of change from 14-day to baseline was able to classify whether patients received drug or placebo. So the information in ventral striatum and to a lesser extent putamen, there was information that sort of reflected drug assignment. So just to summarize this second part, there is this inflammatory subtype that does seem to be linked to motivational anhedonia. The results, the effects of inflammation on motivated behavior may result from suppression of mesolimbic dopamine. And I think most critically, anti-inflammatory strategies are really showing promise, but only when samples are enriched for inflammation and when clinical endpoints are really oriented towards measuring motivational as opposed to consumatory anhedonia or depression severity more generally. So if we're going to see the benefits of these interventions, we really need to have more aligned clinical endpoints to detect the change. And just illustrating this was a recent meta-analysis focusing on the effects of inflammatory drugs in studies that enriched for high CRP and looked at anhedonia as an endpoint. So with that, I'd like to thank the many people who have sort of contributed to this work in my lab, and thank you all for your attention.
Thank you so much, Michael, for a really wonderful talk. And we have now about 15 minutes for question or discussion. You can just raise your hand and I'll call you, or if you prefer, you can also type in the chat and I'll out your questions. So do you have any questions? Just wait a little bit. Maybe then I'll start with one question. I'm very much interested in age differences, and I just wondered whether you can elaborate a little bit more, whether you also find some differences there that are systematic, or whether you'd say actually with this type of variables, age doesn't explain a lot of differences.
Yeah, no, I appreciate the question. We do include age as a covariate in all our models. We don't see for the most part that age has low effects. We do, however, cap age inclusion at 65 for most studies. And I do think that as you get into older adults, a lot of these processes may accelerate, but I think you need slightly different protocols to measure them appropriately, to differentiate between sort of more vascular forms of depression, non-necessarily inflammation, but age-related reductions in dopaminergic tone that can occur. So I think many of the sort of the principles of the pathophysiology that I outlined are extremely relevant for late life depression. but I think it's hard, at least at the stage the field is at now, to mix those different age groups in single studies.
Right. Any questions, comments? Yeah, Colleen?
Hi, thank you so much for the talk. I have just a general question about inflammation. So you
Yeah.
you choose people who have high inflammation, but is not due to kind of disease or autoimmune conditions or anything. So is it just a kind of general variability? So we don't know what's causing their inflammation. Could it matter what's causing the inflammation? Just
So it's a very good question, and it makes a lot of sense, right? Like if the argument is that inflammation is having this effect on the dopamine system and maybe other things as well, like why should it matter where it comes from? And I think inflammation, what we're actually measuring is a marker of activity of the innate immune system and how engaged it is. And how you interpret that marker is, I think, quite context dependent. If you have an acute infection and you have, a real increase in inflammation, that probably will lead to a temporary reduction in dopamine availability and why we feel sort of fatigued when we're inflamed and we're fighting off an infection. That's part of sickness behavior. But that whole process is generally adaptive. And the presumption is that when the infection remits, the inflammation will go back down. You'll feel energetic again. Um, so we want to rule out, to the extent we can, cases where the immune system may be really appropriately, and it's not necessarily a evidence of a sort of hyperactive immune system. It's also worth noting there are bidirectional influences here. So things that will drive chronic inflammation include, poor sleep, poor diet, poor exercise. These are all things that are commonly associated with depression. Um, so there can be some bi-directional relationships there. But I'd also say, we rule out obvious sources of inflammation, not because we think in no case are they relevant, but again, what we think we're trying to target is the individuals who have a vulnerability to this inflammation. We don't know how to measure that yet. That's really the question in this field is a marker of who's going to be vulnerable. Um, so the recruitment strategy is really around, given that we don't know that, using what we can measure, how do we enrich our sample to be as likely as possible to include people who have that vulnerability. Um, and that may mean we're excluding some people who have the vulnerability and also happen to have inflammation due to an autoimmune condition. But it sort of allows for the cleanest interpretation of the sample we have.
And we have a question by Jennifer Crawford.
Mhm.
Hi, thanks for the talk. My pertaining to bridging the two aspects of your talk, and I was curious if you had mentioned that in a lot of cases, these within-person EMA models, where you're looking at the association between effort and affect, you modeled those random slopes, and I was wondering if you had extracted those random slopes and saw if they were related to the inflammatory markers, any sort of striatal
Yeah,
signaling.
it's a great question, and I would love to do that. Fortunately, in our infliximab trial, we didn't yet have the EMA protocol. Um, we are, for some of these ongoing samples, we are collecting blood on those participants. So that is something we will be able to look at, and it's a great suggestion. We have not. We tend to run all our blood analyses in batches, one at a time, or all together to reduce batch effects. So we're holding off until we're done recruitment, but we'll definitely be high on our list.
Then we have one more question in the chat by Melina. I will read it out. She writes, thank you for a great talk. Do you have any idea about why you see different improvements, changes in mood in different types of anhedonia after effort exercise versus exercise, apparently?
Um, so so are you reading that as after effort and
That
exercise, or is the claim that exercise is behaving differently?
that would be
Okay.
on.
Um, we did separately in a subset of these data look at um, high versus low physical activities specifically. And we do find that individuals with higher MA do engage specifically in not just less effortful activities overall, but less physically demanding activities. Um, I, um, I do think that, I mean, it makes sense that exercise would have a mood elevating effect, particularly for those with motivational anhedonia. The fact that we don't see it for consummatory anhedonia is interesting and is part of what is at least leading my thinking that consummatory anhedonia is really somewhat of a different animal and potentially a lot harder to treat because it's sort of not responding. It doesn't seem to be part of the story. It doesn't, follow the pattern of that what you would expect in terms of responding to, behavioral activation type approaches. So it's there's a lot less, I think, well understood about it.
Thank you. Then we have a question by Corinna Schulz.
Yeah, thank you very much for the interesting talk. I was wondering, in the beginning, you mentioned the EMA results. Anhedonia was related to lower effort, but more high reward choices, and that consummatory anhedonia was more related to more effort choices, but less rewarding. And I don't know if you mentioned it already or I missed it, but I was wondering if high effort options were always correlated with like being more rewarding, or if that could be kind of orthogonal to each other. And also wondering if objective effort also correlated then with the subjectively perceived effort, because I think you showed the results basically for the effort obtained from this independent sample,
Mhm.
but I wonder if you also asked them about how they perceived the
Oh,
effort.
yes, we definitely did both. Um, the and so this is an area that we're still analyzing the data now. Sorry. And what we see the sort of strongest associations between the individuals who do the highest, looking at subjective ratings, individuals who have the highest number of high effort, high reward activities, as compared to high reward, low effort, low reward, high effort, low reward. They also tend to have the highest positive affect, which we think is sort of an intriguing association. We're trying to look at the within subject patterns, like does doing something high effort and high reward really boost positive affect more than, say, even high effort, low reward? There's kind of this growing question in the literature around do we really try and avoid effort as much as people suggest, or is actually doing something that's both rewarding and effortful the thing that people find most rewarding? Ongoing work. But do have subjective ratings for all of the activities in addition to the objective ratings I showed. They do correlate, although they're definitely not objective and subjective. I mean, they do correlate, but it's not so strong. And I do think that it is important to consider that people with higher anhedonia may be engaging in activities that are sort of quote unquote objectively rated as, say, rewarding, but their subjective experience of them is not as rewarding. And that's part of what we're trying to tease apart next.
Thank you. Then we have a question by Julia Vogt in the chat. She writes, really enjoyed the talk. Do you have any insight into whether people remember and or learn when they experienced improvements in mood in response to effort?
Oh, it's a great question. Short answer is no. I don't have data on that. The closest we've kind of come to that is trying to look at the influence of sort of this sort of kind of prediction error like metric of like, you expected something to be this effortful and rewarding, and then 2 hours later, how was it? But that's not really capturing learning and memory dynamics. Um, we are in a sort of subsequent data collections adding a voice diary component once a day where people will sort of in response to an open-ended prompt sort of be able to tell us about their experience throughout the day, and that will be a useful tool to being able to relate like what when asked like, hey, how did you feel today, like what do people recall, what comes to mind versus what were they actually reporting throughout the day. But that's all really future directions for us right now. So it's a great thought though.
Then Amira asks, how did you choose the aid in activities in the EMA study, and how was outcome of the activity operationalized?
So we chose the set of activities through a series of pilot studies done about 8 years ago in the lab, sort of looking at what were modal responses. So I would need to go back and sort of check our notes around those. I'm happy to chat off that. And then for the second part, the operationalization, I mean, all we're doing is we're asking people, we represent them with what they said they were going to do from the prior survey. So that gets kind of piped through to the next survey. You said you were going to do these 3 activities. Did you do them? And it's just on them, their evaluation as to whether it happened or not. We don't have any independent confirmation of that.
Then Sam asks, do you believe that sex differences in immune function might be relevant to characterizing an inflammatory subtype of depression?
Yes, I do. Um, we we have not had as much success as I would really like in getting well-powered samples to test for sex differences. Our samples, I mean, it's sort of telling the samples that meet this inclusion criteria of having high inflammation and depression. They tend in our studies and other literature to be close to 80% female, which is much higher than even the general population, higher prevalence of females to males when it comes to depression. So, and we do see evidence in, say, like our iPSC work, the effects on dopamine derived from iPSCs from female donors are much stronger effects of inflammation on dopamine function as compared to male. So I think we need larger clinical trials that are well powered to look at sex effects, but I strongly think we will find out that this is more common in females as compared to males, or more relevant pathophysiology. Yeah.
Well, and I think with that, we're coming to an end. Thank you so much again, Michael,
Thank you.
for joining us today. It was a real pleasure. Please give him a virtual hand wherever you are. And let me just mention, we'll have a next talk in our series, March 25th, by Steve Chang. Hope many of you can join again. Thanks all for coming. Yeah, and have a wonderful afternoon, remaining morning, whatever. Bye.
Thanks so much.